Welcome to the lecture series in food microbiology. In this session, we are going to talk about plant-based toxicants, that is the natural metabolites. It can be primary or secondary. In most of the cases, it is the secondary metabolites of the plants which they produce for their protection, but that can become a toxin to animals including humans which consume them. Members of the plant kingdom are rich sources of the products of vital importance to members of the animal kingdom, meaning we depend on plants. As you know, plants are the primary producers all over, and we directly depend or indirectly depend on the plants as a source of food. They provide the oxygen, energy, vitamins and shelter that are essential to animal life. They are also a rich source of small molecular weight, non-nutritive natural products often called as phytochemicals. These phytochemicals can exhibit beneficial effects on humans, for example drugs or hormones, or they can have negative effects which we call as phytotoxins on animals including humans. So, these metabolites, which are mostly secondary metabolites, which the plant produce for protection or for multiple reasons, to sustain from stress, to help them to protect themselves. They can be beneficial to human beings. For example, many of the drugs, for example quinine from cinchona, and you had the basics from Salix, you had the first painkiller, the discovery of aspirin came by chance. So like that they became drugs. We still know we have so many secondary metabolites which are now ingredients in many pharmaceutical products, hormones, etc. Or they can also have a negative effect on the animals which feed on them. We call them as the phytotoxins. In some cases, the plant products play roles in the plant's defense against the other organisms. So to prevent them from being eaten by animals, the plants produce these products. An allelochemical, this is a chemical substance that is released by a plant that inhibits the growth or germination of another plant. So for it to grow and sustain, it doesn't want a competition from a similar species or a genera which will utilize the same compounds from the same area. So it will produce a chemical which will inhibit that. That is called as an allelochemical. And this process is called as allelopathy. And a phytoalexin, this term phytoalexin, it is a metabolite that is produced by the plant, usually in response to damage by plant pathogens, and that can inhibit the growth of invading organisms. For example, in the plant kingdom, you have very well known, for example, you have the touch me not folding its wings. So when for the sensory, it is a sensory feature. You touch it, you grow near it, the leaves will fold. Similarly, you have the rose and other plants having thorns, so that the insects will not. So when the cattle comes to bite it, the thorn will hurt the tongue, so it will avoid being eaten. And you have very well known cases where you have when an animal or a herbivore comes and chews or breaks a part of the branch or a leaf, in response to the stress of that bite, the plant will produce certain volatile chemicals. These volatile chemicals, in turn, volatile meaning it spreads through the air. So these volatile chemicals will spread through the air, telling the other nearby plants that this herbivore is coming. So this spreads downwind. So in turn, during the course of evolution, the herbivore also learns. So what downwind, as this chemical signal is interpreted, the other plants will start secreting more and more of the secondary metabolites, which would either cause a disflavor so that the animal will not feed on it, or will give a sort of shock or an unpleasant sensation to the ruminant which is eating it. Okay, so now this animal, if it goes and bites the next plant which is dormant, it will not be able to eat it because of either of the disflavor or of the toxicity. Now, evolutionary, how did the animal adapt to this? It learned not to eat plants downwind. So this is a protection, this is eat and being eaten. This happens during the course of evolution. So the Metabolites are the chemicals which the plant usually produces in response to damage. It is called as a phytoalexin. The first class among them which we are going to discuss is the goitrogens. A goiter is defined as a non-cancerous outgrowth and a non-inflammatory enlargement of the thyroid gland, and this has been recognized since ancient times. The first description of thyroid goiter dates back to China in 2700 BC before Christ. And human goiter related to iodine deficiency is an important health problem in certain parts of the world. And in some areas, consumption of cruciferous plants can be a contributing factor to human endemic goiter. A goiter is produced when the seeds of various brassica species, that is cruciferous plants, are fed as a part of the diet to animals, and it is an adaptive response to insufficient levels of thyroid hormones. In the adult, it is usually non-lethal, but it is readily diagnosed symptom of hypothyroidism. Other more serious effects of hypothyroidism include thyroid dysfunction, the loss of energy, increased cardiovascular disease risk, impaired mental and physical development, which is otherwise called as cretinism, and prenatal and infant mortality. Many plants of the mustard family, otherwise the cruciferae, contain certain thyrotoxic substances such as goitrin, allyl thiourea, and the thiocyanate. For example, the cabbagey smell which you get, or from the cauliflower when you cook it, that is because of these isothiocyanates. These substances are not present in undamaged plant, but they are produced by the enzymatic conversion of the thioglucoside precursors, which are called as the glucosinolates. So, a plant which is not damaged, which is not stressed, will not contain these glucosinolates, but they have the precursor molecules, thioglucosides. These precursor molecules, by enzymatic conversion, get converted into the glucosinolates. So, when the required thioglucosidase comes in contact with the substrate, then you have the disruption of the cell's integrity. Upon enzymatic hydrolysis, the glucosinolate called progoitrin, it is rapidly converted into goitrin. This progoitrin is present most notably in sweets and turnips, and at low levels in other plants of the Brassica genus, including cabbage, broccoli, cauliflower, kale, kohlrabi, Brussels sprouts, rutabaga. That is why almost all plants of this Brassica family, which we consume as a vegetable, they do contain this. This thiocyanate, it is an additional dietary goitrogen of considerable importance. Thiocyanate is produced as a byproduct of the glucosinolate hydrolysis and as the principal detoxification product of cyanide. So, cyanide gets converted into thiocyanate, and the glucosinolate hydrolysis also gets formed into thiocyanate. This thiocyanate, so produced by either of the pathways, will interfere with the active uptake and the concentration of inorganic iodine by the thyroid. Remember, iodine is very important for thyroid function. So, this thiocyanate will go and interfere with the concentration, uptake and concentration of the iodine by the thyroid, and it will also inhibit the enzyme thyroperoxidase, thereby preventing the incorporation of iodine into the thyroglobulin. And several staple foods, very important food in the tropics, contain large amounts of the cyanogenic glycosides that are detoxified as thiocyanate. So, you do not have to literally consume thiocyanate. You have the precursor molecules which you feed. For example, cassava contains this, yam contains it. So, they get converted into this thiocyanate, which in turn can harm you. And these plants include cassava, millet, yam, sweet potato, corn, bamboo shoots and lima beans. A further important group of naturally occurring antithyroid substances are the plant polyphenols. Chemicals in this large class of natural products have diverse roles in the plants, ranging from protection against UV light, pathogens to providing colors that attract pollinators. UV, as you know, is a mutagen. It normally causes the thymine to dimerize, and in many cases intensive dimerization will stop DNA replication because the repair doesn't happen because it is too much. So, the organism dies. That is the basic principle. So, any organism, and sunlight, UV light is a part of your sunlight, you get exposed to it. So, all of us have repair mechanisms which can help us to sustain that. Like that, plants also produce polyphenols, which is a normal natural protectant against the UV light. These polyphenols have a secondary function as a protectant against pathogens. And in addition, many of the polyphenols are colored. So, these color will attract the pollinators. For example, you have the quercetin, the resveratrol, and the epigallocatechin produce many positive effects. So, many of these polyphenols act as antioxidants also. So, they have so many positive advantages to the people who consume them. Another important type of disorder which humans get by consumption of certain pro-oxidant chemicals, it is called as favism. This is a hemolytic disorder, meaning RBCs get broken down. In genetically susceptible population exposed to pro-oxidant chemicals, including those which are found in the fava beans, otherwise called as Vishya fava. So, this is a genetically, this is not spread throughout, all humans do not exhibit this, but certain group of people have the genetic susceptibility, meaning their chromosomal makeup is in such a way that they would not be able to break down the toxic product which is found, the pro-oxidant chemical which is found in the fava beans, Vishya fava, and get it broken down into a metabolite which is non-toxic. So, instead of that, they show the symptoms which are hemolytic anemia, jaundice, and hyperbilirubinemia. So, normal people will not be affected, but the genetically susceptible people exposed to these chemicals would show the symptoms of favism. And why genetically susceptible? This is because in these people who show this disorder of favism, they have a deficiency of the erythrocyte glucose 6-phosphate dehydrogenase, an enzyme which is called as G6PD. So, why does this happen? This G6PD gene, the enzyme is coded by a gene. That gene in turn, it is a mutation in the X, that is this particular G6PD is present in the X chromosome. Okay, if you have mutations in that area of the X chromosome which contains this G6PD, then it will manifest as this disease. This is a result of mutation in the X-linked G6PD gene. This is called as human enzymopathies. You have a group of disorders which are produced in human beings because of lack or deficiency of certain specific enzymes. The common term for such disorders are enzymopathies. So, G6PD is a common enzymopathy, and this is approximately present in 400 million people throughout the world. It is more prevalent, however, in areas of Africa, India, in the Mediterranean Sea, China, and the tropics. And why? Why this is highly prevalent in these areas? Because these tropical areas are endemic to the malarial parasite. And remember, malarial parasite multiplies in RBC. So, if RBC is mutated, for example, you might have heard that sickle cell anemia people, where your RBC instead of your normal shape, it is sickle shape, they are resistant, they are not susceptible to malaria. So, that is an adaptation. They would be anemic, but they would not fall sick of malaria. So, like that, here in the tropics, which is a malarial endemic area, you have high G6PD deficiency because as an evolutionary response to the malarial parasite, especially the falciparum malaria, which is very dangerous, Plasmodium falciparum, against which G6PD deficiency plays a protective role. Falciparum malaria is dangerous because the parasite is able to cross the blood-brain barrier and get into the brain. Normal parasites do not do this. So, in the area endemic to this Plasmodium falciparum, you have more evolutionary presence of, or you have a genetic population which contain the G6PD deficiency, which in turn will protect them from attack by these malarial parasites. And when scientists analyzed the bean for substances that decrease the levels of the reduced glutathione in the isolated erythrocytes, they led to the identification of two related glycosides, visine and convisine. So, what led to the development of the symptoms was the occurrence of two glycosides, the visine and the convisine, which was present in the bean. G6PD deficient people were not able to convert this, so it resulted in the abnormality. And cooking does not appreciably reduce the levels of the glycosides in the beans. Hydrolysis of glycosides by exposure to stimulated gastric juice or to beta glucosidase, which is present in high levels of ripe beans, produces the active glycans, the divosine and the isoramyl. So, what do you get? You have the hydrolysis of the glycosides either by exposure to gastric juice or by exposure to beta glucosidase, and in turn it will get converted into glycans, the divosine and the isoramyl. And a further phenolic substance that has been implicated in the cause of favism is L-dopa. It is otherwise called as 3,4-dihydroxyl L-phenylalanine. This L-dopa, it is not active by itself in producing the effects of favism. So, alone it will not cause the symptoms of favism, but when L-dopa combines with this divosine and isoramyl, the presence of all the three combined together will have what we call as a synergistic effect, which in turn will increase the hemolytic activity. The next set of disorder which we are going to talk about is neuroletharism. This in turn is a disease which is caused by the consumption of peas, Letharis sativus, sweet peas. This neuroletharism is a progressive neurodegenerative disorder where the motor neurons get degenerated, and it is characterized in humans, especially in men, by a stiffening of the muscles in the legs that predominantly target a type of neuron which is called as the Betz cell. So, the spinal tract has a type of neurons which is called as the Betz cell, and in neuroletharism, where the toxin produced by the pea plant affects people, it is this Betz cells which get degenerated, thereby causing the symptom of the disease. The initial symptoms are increasing rigidity of the calf muscles and loss of control over the legs. The toxin in the peas that causes the disorder is the amino acid derivative of beta N oxalyl AB diaminopropionic acid, which is otherwise called as the ODAP. The third class of secondary metabolites which we are concerned with are the cyanogenic glycosides. The cyanogenic glycosides, they are a group of water soluble substances that occur naturally in many plants, and more than 2,500 to 3,000 plants have been identified which contain these cyanogenic glycosides. And common plant products that are rich sources of cyanogenic glycosides include lima beans, sorghum, bitter almonds, choke, cherry seeds, apple seeds, etc. All these contain cyanogenic glycosides. So, the plants have evolved these cyanogenic glycosides to protect the seeds from being eaten by foraging animals so that the species can sustain. Cyanogenic glycosides can function in the defense of many of these plants against the invading pathogenic organisms and against many herbivores including humans. Cassava root is a major source of dietary carbohydrate for hundreds and millions of people in South America and Africa. The levels of the cyanide which is available from unprocessed cassava are rich enough to produce serious effects of cyanide poisoning on chronic consumption. So, cassava is a basic carbohydrate source in many, especially in areas which are poverty ridden, but they do contain the negative aspect of cassava consumption is the high presence of high amount of cyanide. Detoxification of the cassava, it is a multi-step process that includes chopping and grinding in running water, which induces enzymatic production of hydrogen cyanide, and it removes both the freed cyanide and the glycoside in water. So, chopping and grinding and washing in running water will remove the cyanides, making the product safe to consume. The symptoms of acute cyanide intoxication are mental confusion, muscular paralysis and respiratory failure. The next class of compounds which we are concerned with are the lectins. These lectins are a group of complex compounds which are widely occurring proteins and glycoproteins which can bind to certain carbohydrates. So, lectins are proteins or specifically glycoproteins. They can bind to certain carbohydrates, and these proteins normally can contribute to the defense of the plants against insects and herbivory by other organisms, including anti-nematode properties either in vitro or in vivo, and lectins are normally toxic to animals. Lectin binding to carbohydrate components of the cell wall can cause clumping, or otherwise what you call as agglutination. This agglutination property is actually used in science as a basis for the assays of RBC blood types. That is, we call type A, type B, type O. That RBO blood typing, ABO blood grouping is because of the property of agglutination. When lectins bind to carbohydrate components and intestinal epithelial cells, the result may be decreased absorption of nutrients from the digestive tract. Some lectins, especially those like resin from the seeds of the castor oil plant, Ricinus communis, and abrin from the jacquerie bean or the rosary pea, Abrus precatorius, they are serious poisons. So, some lectins are mild and they may just impair or lessen the nutrition absorption from digestive food. But you have lectins like the resin which are extreme toxins, serious poison. So, what happens when you consume resin? This causes severe necrosis of the intestinal cell wall and death via multi-organ damage. So, what happens when you consume resin? It is a multiple subunit protein. So, the longer subunit B with the lectin properties will bind to the galactose group of a glycoprotein or a glycolipid which is present on the outer side of the mammalian cell. So, you have this B subunit of the lectin getting bound to a glycolipid which is present on the mammalian cell. So, now this binding in turn will cause an endocytosis. So, the whole complex is taken into the cell inside a vacuole. So, once inside the digestive vacuole, the resin molecule which was so far bound will be released from the glycoconjugate molecule, and subunit A and B are separate. So, these will now separate out. It will be released, it will break down into the components by breaking the disulfide bond. The B chain of the resin makes a channel through the vacuole wall. The A chain will enter the cytoplasm, and upon reaching the ribosome, it will block protein synthesis and kill the cell. So, what we saw was that once consumed inside the cell or once taken it inside the cell by endocytosis, the molecule is released. It will break down, it will break the disulfide bridge, the components will separate, the B chain will go and make a hole through the vacuole wall, and then through this hole the A chain will enter inside, and once it reaches the ribosome, it will stop or inhibit protein synthesis, thereby killing the cell cancer. The next class of compounds which we are going to talk about are the vasoactive amines. They are small molecular weight basic organic compounds which can elicit fight or flight response in higher organisms. In response to the appropriate stimulus, neurons in this system release primarily non-epinephrine and epinephrine. This is also called as noradrenaline and adrenaline. So, neurons will start secreting epinephrine and norepinephrine, and this released epinephrine and norepinephrine in turn will go and bind to the adrenergic receptors in the peripheral tissues. So, along with dopamine, these are structurally related catecholamines. And along with this group, you have additionally the serotonin and tyramine. So, activation of these adrenergic pathways by either dopamine or the catecholamines or the serotonin or the tyramine, any of these will induce the characteristic responses that will include dilation of the eye pupil, increased sweating, increased heart rate, occasional vomiting, and increased BP, blood pressure. Caffeine. This is something which most people are familiar with. Ingredient in coffee. It is a methylated xanthan derivative which occurs in very high amounts in coffee, tea, and chocolate products. The discovery of caffeine containing plants are ancient. People from long ago knew this, and the associated legends are part of folklore of many countries or many cultures. Archaeological evidence indicates that our human ancestors may have consumed caffeine containing plants as far as back from the time 3500 before Christ. And early people found that chewing the seeds, bark, or leaves of certain plants had the effects of easing fatigue. So, it gives you some relaxation. You will no longer feel tired, stimulating awareness and elevating the mood. A popular Chinese legend claims that an emperor who reigned in about 3000 BCE accidentally discovered this, that when some leaves, this is thought to be tea leaves, accidentally fell into boiling water, you had the release of a fragrant aroma, and the drink so formed had proved to be restorative. So, it was an accidental discovery, and the history of coffee has been recorded as far as back from the 9th century BC. Caffeine, as you know, is a neurological stimulant that produces biological effects in almost all organs of the body. The major cellular function of caffeine is to block the receptors of adenosine, and this will act as a negative regulator of function throughout the body. Blockage of these receptors by caffeine and the related purine, theophylline, and therefore stimulates the activities of the associated organs and the tissues, including the central nervous system. So, caffeine becomes a stimulant, but in higher doses this can also act as an addictant. So, low dose would be 200 milligram for an adult, and in this case it will produce central nervous system stimulation, but the toxic effect would include nervousness, irritability, and cardiac arrhythmia. So far we have discussed a couple of natural toxins which are present in the plants. Some in mild doses act as curatives. In higher doses they can become dangerous. We will continue more about these toxins in the next couple of lectures. Thank you.